The liver is a chemical factory essential for human life. Of all the world's pathogens, only 5 viruses have evolved to target it exclusively. While hepatitis B, C, and D typically infiltrate the body directly through the bloodstream, hepatitis A and E take an entirely different route. They enter right through the front door, the human mouth. This is known as enteric, or oral fecal transmission. It relies on a host unwittingly ingesting microscopic viral particles hidden in contaminated water or food. To survive being eaten, these 2 viruses share a specific biological advantage. They are non-enveloped. They lack the fragile outer lipid layer that many other pathogens rely on. Because of this, they are heavily armored, capable of brushing off environmental hazards, industrial cleaning agents, and the highly acidic bath of the human stomach. Because they lack that single outer envelope, hepatitis A and E endure the journey from the outside environment all the way through our digestive tract. They turn the basic, necessary acts of eating and drinking into a global vector for disease. Once these armored viruses survive the stomach acid, they begin to multiply in the intestines. From there, they use the bloodstream as a highway to their ultimate targets, the liver cells, known as hepatocytes. In this diagram of the viral replication cycle, the virus attaches to cell receptors, releasing its RNA into the cytoplasm. The host cell reads this foreign RNA, manufacturing a continuous viral polyprotein that is chopped into parts. These are assembled into fresh viral particles and finally released, destroying the host cell. The virus relies on a specific tactical window. Long before the host ever feels sick, millions of these newly minted viruses are dumped heavily into the bile and subsequently into the feces. Because of this delayed symptom onset, a person will go about their daily life unaware they are infected. They act as an asymptomatic biological factory, distributing the virus back into the environment. At this point, the paths of hepatitis A and E diverge. Hepatitis A relies exclusively on human hosts, which means its spread is directly tied to national sanitation infrastructure. This map shows the global prevalence of hepatitis A. In developing nations, the deep red zones, children are immunized naturally early in life. In highly developed nations, the gray zones, the virus rarely circulates. This leaves unexposed adults vulnerable if they travel to high endemicity areas. Hepatitis E genotypes 1 and 2 follow this same human-to-human pattern, but genotypes 3 and 4 operate under an entirely different set of rules in wealthy, industrialized nations. These specific genotypes are considered a zoonosis. A zoonosis is an infectious disease that originates in an animal reservoir, in this case, domestic pigs, wild boars, and even rabbits, before making the jump to humans. So in higher-income countries, people rarely contract hepatitis E from contaminated drinking water. Instead, the infection usually comes from eating undercooked, infected meat or through direct occupational contact with the animals themselves. While often benign, hepatitis E carries a specific clinical danger. If a pregnant woman contracts the virus during her 3rd trimester, it can trigger acute liver failure, resulting in a 20 to 25% mortality rate. Your geographic location, your local plumbing, and your culinary habits dictate which of these invisible viral threats you are most likely to face. They are twin viruses, but they exploit entirely different vulnerabilities in human society. Because the virus flushes from the stool relatively quickly, doctors rarely hunt for the virus itself. Instead, they look for serology, the specific antibodies the immune system leaves behind after fighting off an invader. This chart tracks biological markers over time. The red line, IgM antibodies, shows the patient is fighting an active infection. The light blue line tracks IgG antibodies, signifying past infection or vaccination, granting lifelong immunity. There is a crucial exception. For immunocompromised patients whose antibody responses are too weak to measure, doctors must use RT-PCR, a genetic test that hunts directly for the viral RNA, to confirm the diagnosis. Reading these markers, a simple blood draw becomes a precise chronological map of our viral encounters, allowing medicine to separate a present danger from a past conquered threat. Because these enteric infections usually resolve spontaneously, specific antiviral treatments are rare, typically reserved only for the most severe cases of hepatitis E. The true front line of defense is purely preventative. It requires strict personal habits, like rigorous hand washing, combined with sustained collective investments in wastewater treatment and municipal sanitation networks. To block the zoonotic strain of hepatitis E in developed nations, the defense is behavioral, ensuring thorough cooking of pork and wild game. The final layer of defense relies on vaccines. A highly effective hepatitis A vaccine exists and is strongly recommended for anyone traveling to endemic regions. A hepatitis E vaccine has also been developed, though currently it is only available in China. Hepatitis A and E are incredibly resilient, invisible threats, but ultimately they are defeated not by high-tech hospital interventions, but by the walls of modern plumbing, everyday hygiene, and safe food practices.